Hello, my name is Prashant Hariharan. I'm a PhD candidate in the Department of Biomedical Engineering at Wayne State University in Detroit, Michigan, and a recipient of the Graduate Fellowship from the International Foundation for Ethical Research. The aim of my project is to develop and validate an organ-on-a-chip model of the choroid plexus, the part of the brain that secretes cerebrospinal fluid. An organ-on-a-chip model can be likened to a microscopic diorama constructed using cells. My objective is to advance the understanding of how the choroid plexus performs its barrier and secretory functions. My long-term goal is to treat hydrocephalus, a neurological condition characterized by the buildup of cerebrospinal fluid in the ventricles of the brain, causing an increase in pressure inside the skull. The current gold standard of treatment is the placement of a ventricular shunt that drains fluids from the ventricles. While this approach improves symptoms, it does not address the underlying causes of hydrocephalus, and it suffers from a high failure rate, with up to 85% of shunts failing within 10 years of placement. No non-surgical interventions exist because the mechanism by which the choroid plexus secretes cerebrospinal fluid is not completely understood. My inspiration for this project comes partly from work published by Dr. Hannah Botfield's team at the University of Birmingham. They demonstrated how a drug used to treat type 2 diabetes and weight loss could be repurposed to treat hydrocephalus. This drew my attention to developing pharmaceutical approaches to mitigating hydrocephalus. I have also been influenced by the emergence of organ-on-a-chip models of several different tissue types over the past decade. I was especially captivated by models like the first lung-on-a-chip and gut-on-a-chip that could perform organ-level functions. Being an engineer by training, I was instantly attracted to organ-on-a-chip development because it was a nexus for many different skill types like electronics, coding, biology, physics, and newer technologies like 3D printing. Presently, researchers use animals ranging from mice and rats to ferrets and pigs. In addition to ethical concerns of using these animals, these models typically involve performing challenging surgical procedures that are difficult to reproduce, and the results from these models cannot be easily translated to humans. Moreover, the time and cost associated with husbandry makes it unsustainable to have larger sample sizes. On the other hand, non-animal models that are currently available to us do not capture the complexity of the choroid plexus. Thus, there is a critical need for a robust, reproducible, human-relevant model of the choroid plexus to study the process of cerebrospinal fluid secretion, especially in the context of hydrocephalus. To address this need, I have built an organ-on-a-chip model of the choroid plexus. Using a 3D printing-based rapid prototyping technique, I have fabricated microfluidic molds and cast silicone chips that are capable of generating physiologic shear when perfused. I have successfully cultured a monolayer of human choroid plexus epithelial cells on the chip, and I'm currently in the process of validating my model against previous in vitro and in vivo models by using parameters such as transepithelial electrical resistance, expression of junctional proteins, and selective permeability of specific molecules across the membrane. Once this model is validated, I will apply it to ask questions about how inflammation affects CSF secretion and barrier function of the choroid plexus. With the second consecutive year of funding from the IFER Fellowship, I have been able to purchase reagents and equipment that are critical to the success of my project. This has allowed me to complete the development and start the validation of my model. The generosity of the readers who donate to IFER has freed me from worrying about the budget of my project and has given me independence and flexibility in planning experiments. What sets organ-on-a-chip models apart from previous model systems is that it uses human cells in a dynamic setting, which means that the cells are continuously provided with biochemical and mechanical signals to mimic the natural environment of the body. Such a system persuades the cells to begin performing their normal biological functions outside their natural environment. In vitro models, like the organ on a chip, can give researchers access to tissues that are otherwise hard to reach. Moreover, the cost of manufacture of an organ on a chip is only a fraction of the price of an animal model, which means that for the same cost, a much larger number of samples can be tested. My mentors are Dr. Carolyn Harris and Dr. Bonnie Blazer-Yost. Dr. Harris is an expert on development of in vitro model systems, and she helps me design and troubleshoot my model. Dr. Blazer-Yost is an expert on transport physiology and helps me understand the intricacies of the choroid plexus and its secretory function. Together, they help me design experiments that bridge biology and engineering. After completing my PhD program, I plan on securing a postdoctoral research position with a lab that specializes in organoids or microphysiological and organ-on-chip systems. I would like to add complexity to my model by learning to incorporate human stem cell-derived cells into the chip. In the long term, I would like to start a lab of my own where I will apply my knowledge of organ-on-a-chip systems to studying cancer cell migration and delivery of cancer therapeutics across the blood-brain and blood-cerebrospinal fluid barrier. Being recognized by IFER is of great prestige to me and will empower me to pursue a career in academic research. I'm very grateful for a second year of generous support from the International Foundation for Ethical Research, and I am excited to make my contribution.